persistence of Lyme disease, Spirochetes, an unsolved problem. And this is Dr. Natasha Rudenko. And I won't go into her CV once again. You can actually take a look at it yourself. But we've had Natasha here before, and she's a laboratory science, and a lot of her work is looking at persistence and very good science on persistence. So the theme of this afternoon session from her work from the Czech Republic will be talking about persistence of Borrelia from her laboratory work. Okay, so welcome. Thank you.
Good afternoon to everyone. My name is Natasha Rudenko. Thanks a lot for the invitation. I was actually surprised because I was invited for the third time to speak on the third conference. I think that there is always something new that Jack and Christian think is valuable for this field. I work with Lyme disease spirochete for almost 30 years, and I've been absolutely fascinated with this creature. And a little bit later, I realized that working in the field of tick-borne disease, and especially Lyme disease, is absolutely not a lottery-winning thing. Because the huge controversy that surrounds Lyme disease particularly, and tick-borne diseases in general, is huge. So what we all face is just ignorance, arrogance, the deep lack of funding, and very often offenses like personal, so it's scientific. And for me, year 2013 was kind of memorable one, because in Boston there was a 13th International Conference of Lyme Borreliosis, and there were quite a few events that I really would like to mention. First of all, it was a huge surprise that the first speaker on the conference read the list of 30 names. There were the names of giants from the tick field research, from Lyme disease field research, people who stopped the active work in the science due to retirement or due to the death. And there were really, the contribution to the field was gigantic, absolutely. It was very sad. But then another sad news came, or maybe not sad news, but it was shocking news. The CDC officially released the dates that actually the number of the diagnosed cases in the United States were more than 10 times higher than considered for long years. It wasn't 30,000, but it was more than 350,000. And then the Europe reacted immediately, and it was released that it's actually not the 80,000 in Europe, but 850,000 new cases in Europe. And then when more data released, those numbers definitely overjumped 1 million only diagnosed cases during the year. And the normal person, what would be the reaction? Oh, no, how it's possible? And it should really evoke a medical community, a scientific community, wild public, and the government should release a huge money because there was some kind of silent epidemic that was finally recognized. And guess what happened? Nothing. Absolutely nothing. And I think that actually that was some kind of a game, because the data were widely known by everybody, and they were just officially recognized. It's really hard to believe that people with or without background in tick and tick-borne disease can really trust the information that the widely distributed around the world Borrelia burgdorferi sensulata complex that at the moment represents 23 named and recognized species and millions of unnamed yet because they're not studied deep enough. And out of those 23, 10, not one sensu stricta globally, not 3 sensu stricta Gariniae afzelii in Europe, but 10 species were either detected in humans or isolated from humans It means that in the one or the other way, those species have pathogenic and infectivity sufficient enough to cause the disease that we're dealing with right now. And actually, the ability of spirochetes to adapt and to survive is extremely, extremely amazing. Generally, back to the past, it was already known that well established that sometimes microorganisms can, under certain conditions, be deprived of all visible signs of life, and yet those organisms are not dead. When the original conditions are restored, they can return to normal life and activity. Very clear. Bacterial population in both batch and continuous culture are much more heterogeneous than is normally assumed. And such cultures may consist of several types of subpopulations simultaneously differing in viability, activity, and integrity of the cells. When we're talking about live and death, there is no definite point that you can really say those organisms are alive and those organisms are dead, because in between, there are always multiple populations, even if they are originated from the same cell. They differ in their viability, in their persistence, in their capability to survive or to die. So the return point from Lyme to death or from death to life is definitely unknown. The survival of spirochetes from the Borrelia burgdorferi sensillata complex in a hostile environment is achieved by the regulation of differential gene expression in response to changes in temperature, salts, nutrients, and all other millions of factors. And actually, those changes lead to formation of dormant subpopulation of cells. From the other side, alternation in the level of gene expression in response to antibiotic pressure leads to the establishment of persistent populations. Both subpopulations represent the cell in different physiological states. Dormancy and persistency do share some similarities. Both represent cells with low metabolic activity that can exist for extended periods of time without replication. Both constitute populations with different gene expression profile, and both differ significantly from replicative forms of spirochetes. Persisters are elusive, present in low numbers, morphologically heterogeneous, multi-drug tolerant, and then they can change with environment. The term persistent first appeared during the Second World War. It was Dr. Gladys Hobby who was working with penicillin and streptococcal culture, and in 1942 she found that penicillin, that is recognized the most efficient weapon during the Second World War because it saved a lot of lives. It kills 99% of streptococcal culture, but it leaves 1% of population absolutely intact. That 1% didn't react on penicillin at all. 2 years later, Professor Joseph Bigger gave the name persisters to this subpopulation, and the original definition of persistent was small population of dormant or non-growing bacteria that have not heritable tolerance to penicillin, but have a capacity to regrow and remain susceptible to the same antibiotic. Something like this you have already seen. Oh, I'm not sure. Okay. So if we have a bulk population and antibiotic pressure actually leads to the really fast death. Antibiotics kill fast. If they don't kill, they will not kill ever. And then at some point, only persisters will survive. If you remove antibiotic pressure, those persisters will turn back to this bulk population, the population of the cells that will be sensitive to the same antibiotic again. So it means this is absolutely a chain closed circle that will be interrupted only if other antibiotics that are effective against persistence will be used. This is a scheme that Ingzam applied for another thing, but it is very much truth for the Lyme disease as well. When you see for the gradient of this color, this is an active disease and this is latent disease. So acute infection and chronic infection. The gradient of color show you the gradient in the population. the cells are different. They're different in the speed of their replication. They're different in their responses, but those are alive. Those are causing the disease. But inside them are always a small group of cells that do nothing. They just play dead. They don't grow, they don't die, they just relax and wait for the time. And when this group of population turns to the latent or chronic infection, the same gradient is actually, you can follow the same gradient as well. The population is not heterogeneous. And in the population of those persisters, there are always reverters, or cells that, being silent for some time, will cause the relapse of the disease without any additional factor involved, just because they programmed to do this. If you talk about dormant and persistent cells, the alternative morphology for Borrelia, the best recognized are actually cysts and biofilms. When you look at this picture, this image, it actually shows you extreme house disorder. Believe me, this is not. This is the reaction of spirochete, very happy spirochete culture to external stress. And this is the way how different cells, the different population of cells react on this. It doesn't matter if this stress is antibiotic treatment, or if the stress is the response to the host complement, or this stress is just an immediate starvation process, which is you're forming right now. So there are some cells that just like those, they're unable to protect, and all of them are getting the same signal. All of them are getting the same order. Emergency, survive. But the speed of reaction of every cell is different. So that's why you can see probably the dead cells right here with a dropping membrane. You can see those that are reacting slower, but you can see those smart ones that starting to develop the forms that can protect them from killing, from death. Those are cystic forms that appear under the pressure. And this is actually the alternative morphology that in majority of cases can secure this spirochete survival. When you talk about biofilms, this is a little bit different process. And we think that the biofilm is not a reaction to immediate stress. The cysts are. But in case of biofilm, it needs time. And from a single spirochetes, you can see the formation of small groups that finally lead to appearance of very strange structure. This is the leaf structure. The spirochete, if you have enough time and strong eyes, you can see the spirochete going inside and leaving back. That state is not actually as stable as we think. But another interesting thing is that not each and every Borrelia species, not each and every strain of Borrelia species is able to form biofilms. And working in the lab, we actually found quite a lot of strains, and it doesn't matter. Either they were originated from tick vector or they were originated from rodent host. They were just different in capability to form biofilms. So, and this community is extremely protective against eradication by antibiotic. So what is that? For us, it was always a question. How different are those isoforms of a spirochete? How different are active replicating form and biofilms at cyst in protein and genomic level and gene level? Actually, so we decided to do analysis, proteomic analysis and transcriptomic analysis, and that was the beginning. We started from the end. We decided to work with proteomic first. And for comparative proteomic study, we actually got a triplicates of active replicating forms, biofilms and cysts. And after that, we separated them into membrane proteins and soluble proteins. And we just ran this analysis, finding that 270 proteins were overexpressed in the membrane fraction, and 397 proteins were identified in a soluble fraction. And you can see some differences in the pattern. So there is active forms, it's just A, B, C, active biofilm cyst. Actually, active, it means replicating. And the soluble as well. 3 active, 3 biofilms, and 3 cysts. Proteome analysis clearly identified the different profiles for each and every population. And cysts appeared to be the most distinctive form, while biofilms and active forms were more or less similar. Pairwise analysis show 149, 282, and 247 differentially expressed proteins for the pairs. This is replicating biofilms, replicating cysts, and those are differences between cysts and biofilms. Red dots represent the downregulated, and the green one upregulated proteins, respectively. Going to the transcriptomic analysis, we found a little bit different picture. Even though the triplicates show the population were grouped reasonably well, we used a replicating form like a cutoff from transcriptomic analysis. We just removed, at this point, we didn't care what's going in active replicating form. We were thinking about persistent or dormant form, something that differs, something that actually rules or give an order to spirochete to go from that happy living, growing, multiplying spirochete to the form that actually it may be happy as well, but not as much growing and not as much replicating. So we found a couple of interesting population and overlap. In those forms, we found 114 genes that were upregulated in cysts and 99 genes that were upregulated in biofilms. And out of this number, 36 were upregulated in both persistent and dormant forms, but not in the replicating forms. And there were downregulation 106 in cyst and 77 in biofilms, and the overlap was 15. And another interesting group, it was 6 genes that were upregulated in biofilm and downregulated in cyst. So actually, we divided all those population to 8. And out of those 8, 4 were of our interest. So the 8 population, it was like genes which expression upregulated exclusively in cyst, exclusively in biofilms, or genes that was upregulated in both as cyst, so as biofilm. The same is downregulated in cyst, only in biofilms, and overlap. And right here, there is upregulation in one form and downregulation in the other, and the same as well. We all know that the mechanism of protein and gene expression regulated by different mechanisms. And whatever happens on proteomic level doesn't reflect 100% whatever goes on genomic level. So that's why we decided to overlap the results of proteomics and transcriptomics and eliminate as much as possible those may be interesting candidates, but that we are not interested in a moment, and to go to those that are really differentially expressed. And from the almost 400 that were identified at the beginning, we actually eliminated it to 113. But things are not so easy as it sounds. When you check this heat map of membrane proteins, and you can see the differences in protein expression, active forms, biofilms, and cyst. And presumably, it's supposed to be easy. You're just picking the candidate with where all differences are shown, and you're getting a unique marker for the specific form. But as you see, hundreds and hundreds of PCRs, RT-PCR shows that actually on a gene level, it's completely different. And knowing the biological function, cell compartment, you cannot use only the proteomics results to define those things. Proteomics and transcriptomics provide supplementing each other results, but they're not identical. And it takes quite a while to find at least something. So this is just a proof of concept that it works. This is one of our so-called candidate proteins that we defined that was overexpressed only in biofilms, but wasn't detected in either cyst or replicating forms. This candidate, this protein, wasn't characterized in Borrelia, but the information that was provided from the literature, and actually what we were able to find, proves that the protein has a capacity to function as either a kinase or as phosphatase inhibitor. That protein was found to bind to the surface of immune cells, but not to the epithelial cell. That this protein has an immunosuppressive role, inhibiting innate immune response in affected human macrophages, and so on, so on, so on. But this is an information that was not based on Borrelia sequences. At the moment, this is unpublished results yet. We did find a couple of unique for a pair active cyst candidates, as well as we found a marker specific for replicating forms that were not present in cyst and biofilms. Only one for biofilms marker and two for cyst marker. This is the first part of my presentation, the thing that we were doing and are doing normally and permanently. And the second thing, the second part, is not what we're doing normally, but unfortunately, we were asked to help in this case, and I would like to present it to you. Borrelia in human brain, postmortem. Male patient born in 1996. In 2004, he was diagnosed with Lyme disease after contacting doctor because he has a maritime immigrants. Antibiotic treatment was prescribed, but unfortunately, the medical doctors that provided this information don't have is the duration of type of antibiotics that were used at that time. The patient suffered from neurological symptoms, mostly cognitive deficits such as brain fog, reduced psychomotor performance, difficulties with concentration, and processing of visual or auditory stimuli. In 2017, examined in the neurobiology department, had a positive Borrelia serology test in both IgG and IgM in serum, but negative PCR in lumbar puncture. Initiated therapy was started, but it didn't last long. In 2018, anti-Borrelia antibodies were determined again, with borderline positivity for Borrelia-specific IgM and strong positivity for IgG. Serological test for chlamydia, mycoplasma, anaplasma, toxocara, and toxoplasma gondii were negative. The PCR test for the presence of Babesia in peripheral blood was negative. Patient was prescribed to a combination of antimicrobials. In August 2018, the suspected hepatotoxicity was ruled out by some tests in a hospital. In September 2018, the patient was hospitalized with suspicion of the developing mental disorder. The patient was diagnosed with schizotypal and somatomorphic condition and was discharged 3 weeks later. In August 2019, the patient committed suicide, and he left a letter behind expressing the urgent demand to scientists to analyze his brain for presence of Lyme disease spirochetes. Postmortem toxicology and microbiology confirmed the presence of chemicals that proved that before his death, he was taking the medication. Immunoblood analysis of postmortem serum and CSF confirmed borderline IgG activity against VLS-E of Borrelia garenia and Borrelia burgdorferi sensu stricta. The reaction with VLS-E antigens of Afzeli and lipids for garenia and Afzeli were negative. Using immunoblood kit for autoantibodies detection, weak IgG positivity against PL7 antigen and borderline reaction with SRP antigens and histones were detected. As a part of microbiological analysis of postmortem samples, the CSF and brain tissues were used for the cultivation of spirochetes in BSK-H medium, and after 2 months, the results were negative, so all samples were deemed to be culture negative. From this case, 7 different samples were taken, or samples from 7 different brain regions were taken. Temporal lobe, frontal lobe, parental lobe, occipital lobe, and cerebellum, as well as choroid plexus and basal ganglia. Unfortunately, there was no specific order in sample removing, so that was a mess. But the good thing was that there were multiple samples from each and every area, and we had quite something to work with. The samples from each area were divided into 3 parts. First one went to cultivation and were used immediately. The second one that was mentioned to be used for molecular detection was frozen immediately. And the last one that was meant to use for further analysis, like microscopic and so on, were fixed at once. We were running our standard procedure. We started with amplification of FLAB gene and OSP. And actually, I think I still consider that those 2 genes are the most informative for the pilot beginning analysis of any kind of or any species of Borrelia spirochetes. And we were surprised, really, to get a positive result, because the body was found 3 days after the suicidal, and there was quite a high state of autolysis of the tissue, so it was not much to work with. Then, to be sure that we are not mistaken, we applied MLST scheme for amplification, so there were 8 housekeeping genes, and sequencing, at least at the beginning part of the genes, we found interesting thing. 2 species were present in the brain: Borrelia burgdorferi sensu stricta in 5 areas, and Borrelia burgdorferi that we detected presented the strain with OSP type B, which is for Europe the most infective strain. The most infective globally is OSP type A, and this one was B. And 2, let's say, were infected with, we detected the presence of DNA of Borrelia garini in 2 areas, basal ganglia and cerebellum. Interesting thing, first of all, is that this is the first case of detection of 2 species in the human brain. And another thing, that there is no overlap in, there is no overlap in the area where it was detected. So this is actually the area, I had to run, sorry, as always. Those areas that were PCR positive were used for further analysis, and this is images that actually you can see in work of Alan MacDonald, Eva Sappi, Judith McCloskey, Monica Ambers, Catherine Brissett, everyone who works with it. This is a typical presentation of atypical forms of spirochete in human brain that we detected postmortem. We were trying to actually repeat the same case in mice, and this is the presence of S spiral, so as atypical forms in mice tissues. And going further, the spirochetes in mice bladder, the spirochetes in mice joints, and this is the presence of biofilm, and more pictures that confirms you the spirochetes survive antibiotic treatment and are detectable by different techniques in my tissues as well. And the very last slide is persistent eradication. This is not my thoughts. This is ideas of smart people that were published and that I selected from a database. Very few existing antibiotics are active against non-growing stationary cells, and all are ineffective against dormant persisters. Persisters are specialized survivor cells, and with multiple mechanisms of formation, do not provide a good target that would disable their formation. One possible approach is to look at the Achilles heel of persisters, not the mechanism, but rather a protein essential for maintaining the dormant cell. Possible target is a global regulatory PHOU. Combining a regular antibiotic with a compound disabling a persister maintenance function would provide a sterilizing therapeutic. Another possibility is to develop prodrugs compound that enter into the cell and are converted to a generally reactive compound by a bacteria-specific enzyme. The active species will then attack unrelated targets, including DNA and the membrane, and kill dormant cells. And thank you for your attention.